And then I'm gonna go ahead and share my screen. This just takes a little bit of finagling to get the images so I can actually see you guys. Okay, let me put this stuff down real quick. Lecture 1. Okay, so hi guys, welcome to Neuromotor Control. I know a bunch of you guys, so that's really, really nice. I love being able to call y'all out and make sure that everyone's doing okay and that you guys feel totally comfortable with me. For those of you that have had me for motor development or for research methods, this is a totally different kind of class, okay? This class is gonna be a lot more like a traditional anatomy and physiology class, but ratcheted up for a graduate level. So we'll start reading primary articles that builds upon the material for class. And so it's different. It's definitely different. So in any case, let's just kind of get the ball going. So today we're actually going to cover part of a lecture. So we'll try to take the syllabus and our introductions as quickly or slowly as we need to, and then we'll dive into the material. Okay, so today we're going to go through just introductions. Who are you? Who am I? We'll go over some of the syllabus information, and we'll dive into chapter 1. Well, let's get going. Okay, so what I would like you guys to do, we'll just go around the Zoom. Your name, master's or PhD level, what your concentration is, if you're a PhD student, who your PhD supervisor is, if you've ever taken a neuroanatomy, neuroscience, or neurophysiology course in the past, and the last one is just any kind of special skill or talent. We are in neuromotor control, so it's kind of nice to get an idea of the kinds of skills that you guys do, and that could be anything, but ideally it's something in the motor domain so that I have a reference point in terms of what you're able to do. Okay, so we'll just go around the room. Matt, if you want to just put your information in the chat window, and then that would be perfect. Okay, so Daphne, we'll go ahead and start with you. That's okay?
Yeah, for sure. My name's Daphne Schmid. I'm a master's student. My area of concentration is motor control. I have taken a course on the overview of neuroanatomy, but not like anything like super specific, and it was an undergrad. Skills, I play tennis, so that could be one.
Perfect. Yeah, tennis is great. Okay, perfect. Let's see, 1 second. Let's do Taylor. I don't know if you guys can actually see like everyone on here in the same way I do. Go ahead, Taylor.
Hey y'all, I'm Taylor. I'm exactly what Daphne just said. I'm also a master's student. My area of concentration is motor behavior, motor control, except I have not taken a course on neuroanatomy, so I'm really excited. And then I was going to also use that I play tennis. And so Daphne, can we play together? And then, I played volleyball in high school. It's another special skill or talent.
Perfect.
Thank you.
Yeah. I mean, all again, some of these things are going to be, I'm not going to actually make you perform any of them. So don't worry about it. But sometimes it's nice for us to kind of think about when you're doing different types of movements where you have to anticipate like receiving a ball, or when you're serving, making a plan of how you're going to actually hit the ball and where it needs to go. I mean, those require different types of sensory and motor interactions. And so we kind of think about it like that. I know, like, for example, Kylie, you're track and field, right? Yeah. So like Kylie you do hurdles, right? Is that right? Sorry, I'm calling Kylie out just because I remember she's on our track and field team.
I throw javelin.
Oh, you throw javelin. Okay, sorry.
Yeah.
For some reason I thought you were on. Okay, but even then, like there are different types of movements that require different types of processes, and keeping in mind these skills that you have and putting it to the context of these circuits, I think really helps. So sometimes that's really nice to do. So Kylie, since I'm already calling you out, let's do you.
So my name is Kylie, and I'm also a master's student. My area of concentration is biomechanics, and I have not taken a course, like any neuro courses, but I have taken your motor development class, and we talked about, brain areas and stuff. And then my skill or talent, I guess, would be I'm a javelin thrower for the track and field team here. So
Perfect. Malaya.
Hello, my name is Malaya Campbell. I am a master's student and my concentration is in motor behavior as well. I haven't taken a neuroanatomy class, mostly just gone over it in my other anatomy classes in undergrad. And then a special skill talent. Well, I guess I kind of have two. So one is I roller skate, but I also like make my skates. So
Oh, that's awesome. I love that. But you don't do roller derby?
no, no.
Perfect. Thanks, Malaya. Riley.
Good morning, guys. I'm Riley Newton. I'm a master's student in motor behavior, and I have not taken a course on neuroanatomy. So yes, this is very exciting for me. In terms of sports, I don't really have a special skill or talent, but pretty much anything anaerobic, I am decent at and great at nothing. So like, I can run a pretty good 40. I can run a pretty good 40. I can bench a decent bit, do a bunch of pull-ups, but just nothing great in particular.
No worries. No judgment. I gotta feel the same way. I played a bunch of sports throughout high school and learned a bunch of different skills when I was in grad school, but nothing too exciting. Okay, Sean.
Hey, I'm Sean Holmes. I'm a master's student. My area of concentration is motor learning, but I've not taken any like neuroanatomy courses or anything. I'd say a special skill that I have is I can juggle.
Cool. Yeah, you'd be surprised. Juggling, there was a study that was published, I think it was in 2012. We might cover it in class about how learning a new skill, learning a complex motor skill changes specific areas of your brain. And the complex motor skill that they taught all the participants was juggling. And it's associated with all kinds of really good things in terms of brain growth. So you're good to go. Awesome.
My brother can actually juggle 7 balls, and he's like the smartest person I know. So that's something.
So I mean, I definitely think that y'all should take that up. When just as a side note, when I took my first neuroanatomy course, it was kind of like a neuroanatomy and movement disorders class. And one of the first topics we covered was stroke, because stroke can happen and affect all kinds of different areas of the brain. And one of the first things that we were taught was that if you're a lefty or you're ambidextrous, you have a better outcome in terms of stroke rehabilitation, in terms of natural reorganization of your brain. So that entire class and all of the subsequent neuroanatomy courses I took, I wrote with my left hand, even though I'm right-handed. So my notes look terrible, but I was thinking that maybe that might be enough to kind of get the things in a system going. So maybe I'll just challenge y'all to try juggling. We have a bunch of scarves in the lab. So maybe if you guys want to pick up some scarves to practice, we can put that up there. Okay. I can't see everyone's name. So here's what I'm going to do. I'm going to ask you guys to actually turn off your video so I can see your names when it pops up. And then when I call your name out, if that's okay, then I can, you can just turn your video on that way. Is that okay? Sorry, I know it's a little bit annoying. Let's go with Nick.
Good afternoon, everybody. I'm in Georgia, so it's 12 o'clock over here. But
No worries.
my name is Nick Bradley. I'm a master's student with a concentration in motor behavior. I've never taken a neuroanatomy class or anything of that nature. And a special skill I have, I guess, I'm a self-taught musician. So I can play the piano by ear. I can listen to something and I can just sound it out in about 5 or so minutes.
That's very exciting. Yeah, so you're going to be in a similar position. A lot of the work, again, with respect to neuroplasticity has to do with playing music or learning another language. So If you guys are bilingual or you play music or you juggle, you guys are going to be in good shape. But thank you, Nick. Ben.
Hey guys, I'm Ben.
Let me share so you guys can actually see each other's faces. How about that? There we go.
I'm a master's student. I'm also in motor behavior, and I've taken a lot of anatomy classes, but no neuromotor or anything like that specifically. And I can do a pistol squat.
Okay. Yeah, awesome. Are these good? Hold on a second. No, they're gross. What? I'm teaching class right now, Connor. Sorry, my husband's here. Okay, so let's see. Alex.
Sorry, I just like moved because my roommate has COVID, so the dogs are going crazy. I'm all over the place. What's up? I'm Alex. I'm a master's student as well in the motor behavior concentration. I'm an athletic training student, and I was a lacrosse player in undergrad. And I guess my special talent, by no means am I bilingual, but I actually train my dog in German. So I guess switching over from English to German when I speak with him.
That's awesome. Perfect. Sienna.
Hey guys, I'm Sienna. I'm a master's student, also in motor behavior. I haven't taken any neuroanatomy classes. And then I don't know if I really have a special skill, but I really enjoy ultimate frisbee.
Perfect. Great. Charles.
Hey guys, I'm Charles Speaks. In the master's program of physiology of exercise. I haven't taken any, neuroanatomy particularly focused courses, but just, general anatomy courses that go over nervous system and things like that. And I particularly enjoy and like to think I'm pretty decent at golf. So it's my skill.
Perfect. Okay. Anthony.
Hello, everyone. My name is Anthony Fava. I'm first year PhD. I started in the fall. My advisor is Dr. Oliver. We're working in the sports medicine and movement lab. So we're looking at softball and baseball, kinematics, and I've not taken a course specifically for neurophysiology. Special skill, I like to run. So unlike Riley, I'll be your aerobic guy. I like to run distance. I ran a few races past few years. So that's me in a nutshell.
What's the longest distance you've done?
I did a marathon the last 2 years before this past year with COVID.
Awesome. Good for you. That is not something
challenging.
Awesome. Okay, Mariah.
Hi, I'm Mariah. I'm a first year PhD. I started in the fall. My advisor is Dr. Brown. And yes, I have taken a neuroanatomy course. I'm actually a speech language pathologist. So I work with people who have voice, swallowing, and upper airway issues. So I've taken neuroanatomy as it specifically relates to voice and speech sounds. And my special talent is, it's not that great of a talent, but one thing, and if you're working with someone who has like head or neck cancer, who has recently had their larynx removed, one thing we teach them is esophageal speech, and I'm actually really good at it, which is complicated, but something I'm actually pretty good at. So.
How can you give us an example?
It's just, I don't really want to. I mean,
okay, no, that's fine. I don't want to put you on the spot.
Gasp in, and you speak from an inhale instead of an exhale.
Oh, that's interesting. Cool. So Mariah, this class is going to be a little bit different than your previous classes because we're not even covering auditory system at all. And one of the reasons for that is with the exception of vocal motor control and speech language processes, we very much downweight the auditory system in goal-directed movements and often repetitive behaviors that we're doing, like while walking and things like that. And because of that, the auditory system kind of gets relegated to the back. So hopefully this will help to round out some of the other things that you've learned in the past, but definitely keep in mind, I mean, speech production is very much a fine motor control in terms of the number of mappings in terms of moving your mouth, facial muscles, lips, tongue, all of that good stuff. So you can kind of keep that as a context, but this will be a little bit different than the other classes you've done for that reason. Excellent, welcome. Cherise?
All right, can you?
Uh-oh, I'm losing you.
Can you hear me now?
Yeah, we're good now.
Okay. I'm Cherise. I am a master's student in biomechanics. I haven't taken a neuroanatomy class, but I did take motor behavior in undergrad. And my special skill, I guess, would be that I play the ukulele.
Excellent. Excellent, cool. Welcome. Isaiah.
Can you hear me?
Yeah, speak a little louder if you can.
Can you hear me good?
Yep.
Good morning, my name is Isaiah Watson. Master's track, exercise phys. I have not taken any neuroscience or neurophysiology classes, just the basic anatomy courses. And my special skill or talent would be knitting. Since quarantine started last year, started picking up the skill, and I knit probably 2 or 3 scarves a week. That's fine.
Love it. I love that. I knit too. I'm not very good, but I used to do that during football season. I would just watch football and knit scarves for everyone for Christmas. Fantastic. Welcome, Isaiah. Jared.
Hi, I'm Jared. I'm a PhD student in Dr. Weimer's sports biomechanics lab. I have not taken a neuroanatomy or neurophysiology classes before, and my special skill is I can throw a spiral when throwing a football.
Perfect. Awesome. Welcome. Haley?
Okay, can you hear me? Okay. So my name is Haley. I'm a master's student and I'm in the biomechanics track. I haven't taken any neuroanatomy classes specifically, just kind of the general anatomy and phys classes. And my special skill, I guess, I don't really know if it counts, but I enjoy cooking. So I do like a lot of new, like different kind of recipes and that kind of stuff.
That counts for sure. My husband's a chef, so I can appreciate that for sure. And I think Matt, do you want to use your voice to text? Don't put the meat in our face. Connor, please. Okay. I'm going to read Matt's Zoom chat. So Matt is up here. He's a master's student in motor behavior. He has not taken a neuro class. And his special talent is that he's good at doing things one-handed because he has hemiparesis. So for sure, that's definitely something that we'll talk a little bit about in terms of when you do have brain injuries, how you learn to take on new skills or relearn skills with one hand or the other, or with different limbs completely. And so in people that have lower extremity issues or upper extremity issues, there are ways to swap in and out and relearn that as an adult. So Matt's a good example of adult neurogenesis and rehabilitation. Okay, did I miss anybody? I think I got everyone. Okay, just speak up if you guys need anything or need me to stop. I'm gonna go ahead and share my screen again. Okay, so perfect. Okay, so this is me. I did my 2 undergrad degrees, one in philosophy and one in kinesiology, and my master's and PhD all at the University of Maryland. So I'm a Terp for life. I play lacrosse. I've always played lacrosse, field hockey, basically stick sports, ice hockey. My background is in neuroscience and cognitive science. So for those of you guys that saw me or had me in different classes, this is my bread and butter. This is a class that I really love teaching. And I've had a long history of teaching this class. So I'm excited. I did my postdoc at the University of Toronto trying to understand brain development and using MRI and fMRI as an imaging tool. In my master's and PhD, I used electroencephalography and also movement kinematics. And my world is really studying brain injury. I study individuals with developmental disabilities and then also acquired injuries like sports-related concussion. And it's just a fascinating pathway and track. So although this class will not really talk as much about injury as I'd love to, we really need to have a framework for what the typical system looks like in an intact adult before we start talking about what happens in development or in aging or in different types of acquired injuries. Okay, so let's dive into the syllabus just to keep the class moving on. So the learning outcomes for this class is so that you guys will understand sensory motor processes and the different types of motor behaviors, including goal-directed movements, postural control and gait, and interlimb coordination with respect to the neural pathways that underlie those skills. We're going to describe the neural underpinnings of specific deficits in sensory motor processes. Oftentimes this will take the form of again, acquired injuries like spinal cord injury, stroke, or neurodegenerative disorders like Parkinson's disease and Huntington's disease. We're going to critically evaluate some research studies and review papers that employ behavioral, neurophysiological, or neuroimaging techniques to understand sensory motor control, learning, and plasticity. And then a big part of this class is to actually utilize these online tools to gain a deeper understanding of just the clear-cut lecture and classroom material. So there's quite a bit of video lectures, interactive apps, and discussion forums that we're going to take advantage of. Okay, so all the required materials, including the textbook, don't tell the bookstore, is online, available for download, okay? So you can just download the whole thing. I hate that you guys have to pay for materials, especially as grad students when you get paid nothing. So everything's on Canvas. You can download all that. Links, the online lectures and videos are also posted on Canvas. And then any additional materials that I think are interesting, like video interviews, relevant news articles, podcasts, things like that that are relevant, I'll post them on Canvas as well. Okay, so here's a breakdown of the requirements and the evaluation. You will have weekly discussion questions or quizzes. I'll try not to overload you because there is a lot of reading and the material is very dense. But before we move on to the next set of material, you'll have a quiz that's due. Or you'll have a discussion question for the readings of the primary research articles or research reviews. You'll do a group presentation. Usually it's in groups of 2 to 3. We have 19 students in this class, so it's likely going to be groups of 3. I know you guys hate group work. I know it's a pain, especially during COVID. And I know that there are always some members that don't pull their weight. I just really want you guys to work collaboratively on this so that it's a reflection of everyone's efforts. Again, the material that I'm having you guys read is dense. It's very nice to have other people help you to understand the material and for you to get some experience explaining it to other people. Those group presentations, again, if you've had me as a course instructor, I will meet with you guys a week in advance to review your PowerPoints and make sure that you guys understand the material you're presenting. And of course, I'll be there during the presentation, so I'll jump in if there are clarifications that are necessary. Another piece is that you guys will serve as a discussant for 2 of the research articles. What I mean by that is, in addition to posting on the discussion questions on Canvas, you're going to be doing that one in advance of the the presentation, but you're also going to be like the first line in which the group presenters can kind of jump in and say, okay, let's say Haley is the one of the discussants. If I pose a question to the group and no one's responding, then Haley will get the ball rolling to make the presentations a little bit more interactive. Okay, so you'll end up signing up for 2 other papers. Ideally, everyone's going to read all of the papers, but again, they're dense. And I know that you guys have other deadlines for other classes, so just be strategic about which ones you pick. Okay, so then there's that. And then the only real extra credit for this class is if you serve as a discussant for another more than 2 presentations. Again, the goal of being a discussant is that you understand, you really have read the paper as much as you would be presenting it yourself. So it is quite a bit more work, and that will serve as extra credit. And then there are 2 exams, a midterm and a final. The exams take the exact same format as the quizzes, with the exception that they are closed note, closed book, and we'll use honor lock for them. And so the quizzes, open book, open note, open internet. Okay. The goal of the quiz is for you guys to probe yourself to understand the material. You'll have 30 minutes to take a quiz with 10 questions. The goal is for you guys to understand the material, not necessarily for you, just to get the right answer. But the midterms and finals will be closed book, closed note to force you guys to really study the material outside of the quizzes. Okay, the midterm will be the cumulative for section 1, and the final will be the cumulative for section 2. And here's the breakdown. So section 1 will go into studying the central nervous system, which is chapter 1 and the appendix. Intro to spinal cord, brainstem, and cortex covers the appendix. Somatosensory system, vision, and vestibular system will also be covered. In the syllabus, I'll show you the actual chapters from the book and the material that will be linked on Canvas. And starting with vision, we'll start having group presentations and additional reading. So that's the section for the first. And then the second section is going to cover really motor control. So the first section is trying to help you understand the sensory systems that contribute to motor control. The second part is how do we actually control the system? So lower motor control is really lower motor neurons, so spinal cord out. Upper neuron control is going to be cortex and down, and then basal ganglia, cerebellum. Sensory motor integration is going to cover both sensory motor integration at the spinal cord and at the level of cortex. And the last section on plasticity and repair is really going to talk about traumatic injuries and the actual mechanisms underlying central nervous system repair. So the final exam will cover sections 7 through 12. Okay, so again, this class is going to be really dense. It's very much like a traditional neuro or anatomy course or anatomy and physiology course. Make note cards, rewrite your notes, study with a buddy, rewatch my lectures. It's really good to do this along each week and not leave it to the last minute. Come see me if you guys have questions. My office hours are going to be on Wednesdays right after this class until 2. So I'm just going to have open office hours whenever you guys want to drop in, and we can review material as you see fit. Okay, so let's dive into the material. Before I do that, actually, let me stop my share real quick. Let me pull up the actual syllabus so you guys can see some of the details. Okay,
I think the one on Canvas is from 2019.
Yes, I will update that. I will show you the updated syllabus right now. Let me see if I can actually get Zoom to share my screen again. Okay. Okay, so here is the updated syllabus from the current semester. So again, my office hours are going to be set from 12:30 to 2, and it's just through a Zoom link. And then the online material that we're going to use a lot is going to be the University of British Columbia's neuroanatomy course. It is excellent. The videos are really, really well produced. The interactive modules that are on there provide you with a really good idea of what the real anatomy looks like. The Purvis textbook, I think, is really good because it provides really clear drawings of what the anatomy is. But if you actually want to look at what it is in the flesh, like, it's nice to see what's there. So the University of British Columbia program is going to be the most material that we use outside of the textbook. And then Anatomy TV is, let me see if I can pull it up. Give me a second. It's also a really good resource. I really like it. It's called Primal Pictures. Once you click on the link on Canvas, it takes you to an authentication page because you have to be part of Auburn University to be able to access it. There is, let me see if I can find it. Anatomy and Physiology of the Nervous System. There it is. And again, it's very redundant to the material that we have, but it's kind of nice to see like all the different components, to see it in a schematic. It provides pretty good information. You can rotate and like manipulate the material. So if you're more of like a three-dimensional learner, this might be a little bit better for you because you can like literally click on different structures. It'll pull up information. So that's one of the other pieces that is very helpful. Okay.
Look, you take it upstairs.
Yes. Okay. And then the last part is just the MRI Atlas through Harvard. I like this a lot because I actually used it considerably when I was working in an MRI lab and we were trying to understand what different forms of MRI would look like and what the images and structures would look like at different levels. So this is a T2 weighted image. This is a SPECT image. Again, it provides examples in terms of clinical cases. It provides examples in terms of just general anatomy and physiology. So that's great. Okay, so here's, oh, so let me just, sorry, one more thing. COVID obviously is a big issue. If you guys get sick or have any problems, you need to just contact me as soon as possible. Provide me with medical documentation if you can, and we'll do whatever we can to make sure you keep up with the coursework. And if you can't, we'll sort out some way that you can complete the class successfully. Okay, if that means that you need to postpone and take it incomplete, so be it. I'm not super worried about it. Your health is the most important thing. If you experience a mental health issue, let me know. I mean, it's the same kind of situation. Again, your health is very important to me and to the rest of the faculty. Just feel free to reach out if there's something that's preventing you from being successful in the class. If I get sick, the alternative instructors are going to be two of my doctoral students that have taken this class and work with neuroanatomy. So they would be great, and their emails are there. And then again, this is the course structure. I noted when we have health days, or I don't remember, wellness days, and when we have days off. I also noted where there might be some primary readings. I may modify the primary readings depending on what your interests are, and if we feel like we need to actually spend more time in the book material versus the readings. We'll just get a gauge of how you guys are doing across the semester, and then we'll adjust the readings if necessary. Again, I really like to link you guys with outside materials. Sometimes it's nice just to hear the material from someone other than me. Okay, so I'll try and link it to those other things. And then before the midterm and the final, I have optional review days for you guys. So you can ask additional questions and get clarification on any of the material previous to that. Okay. Okay, so that's the syllabus. Let's get back to material. Okay, so one of the really nice things about studying neuroscience is that there are so many different windows into understanding the brain. We have anatomists, embryologists, physiologists, pharmacologists, psychologists, physicists, engineers, computer scientists, and kinesiologists. I added the kinesiology because it wasn't in the book. But all of us provide a different way, either in terms of the actual imaging of the brain, the actual segmentation of the brain, or really trying to understand how the brain works from an engineering or computer science perspective. So all of these different windows have historically provided us with more and more information about what does the brain look like, what does it do, and how does it function. Okay, so there are multiple different ways that we will study the brain in this class, and there's kind of levels of understanding. Okay, so at the most granular or most micro that we'll go is the genetic level. We will not study genetics at that level. We will not talk that much about genes. There are a handful of genes that are associated with specific disorders that we will talk about, but by and large, we will not talk about DNA replication, gene expression, that kind of stuff too much throughout this class. It's honestly above my pay grade. So again, we're not going to get into too much of those details. The same is said in terms of molecular biology and cellular biology. We will not cover a good chunk of that material in the book. And the reason for that is historically, the students that come into this class don't have a strong enough background in physiology and really get stuck in the details at the molecular level. And although it's important to understand the molecular level in terms of different neurotransmitters, how they function within the cell, how they enter the cell, how they're metabolized, most of you aren't going to be interested in that. With that said, the material is there in the book. If you're really interested in understanding synaptic function and specific ways in which the cell processes different types of neurotransmitters or creates neurotransmitters, feel free. But I'm not going to hold you guys to it. What we will focus on in this class is at really at the level of gross anatomy. So anatomy and physiology in terms of dissecting the actual structures themselves through our online material, really, because we don't have an anatomy lab, using neuroimaging, which we've talked about in other classes. So mostly structural MRI, some aspects of functional MRI and diffusion imaging, some aspects, a little bit of PET scanning, but again, we really want to understand the gross anatomy. What do those structures look like? How do they connect with other structures, and what do they do? And then the last part, which I think is really underrated, is a neuropsychological approach. And what I mean by neuropsychological approaches is understanding specific behavioral deficits and the areas of the brain that might be associated with those specific deficits. So a lot of that work comes from the stroke literature or different types of traumatic injuries, or the neurodegenerative disease area. So if we have someone with Alzheimer's pathology, we know that there are specific memory functions that are lost in that pathology. And we know that there are certain regions of the brain that are associated with that pathology. And so we've connected areas of, say, the hippocampus with long-term memory because we have specific patients with damage to that area, and that helps us to link structure and function. So for me, the neuropsychological part is really fascinating because, I mean, a lot of you guys are athletic trainers, some of you are clinicians. Everyone presents with their behavioral deficits a little bit differently. And that's because the way in which their brain takes a traumatic insult or deals with a neurodegenerative disorder is very different. And that individual specificity provides some nuance in terms of the types of behavioral deficits that are exhibited. So personally, I think it's really interesting. I study a lot of autism. Autism is a mess. It's a mess. So, okay. So there are really 3 sets of essential functions in the central nervous system. And we kind of separate them out into sensory systems, motor systems, and association systems. The thing that's kind of nice about neuromotor control is that we have to consider all 3. We'll take part 1 of the class to the midterm to talk about sensory systems. And the point of sensory systems is to get an idea of what the state of the organism and the external environment is. And what I mean by that is, you know, am I sitting or am I standing? Am I horizontal or am I vertical? Am I underwater or am I on land? Am I in the atmosphere? Am I injured or am I operating at full capacity? We're trying to understand how our body is represented in the environment that we're in. And we need that sensory information before we're actually able to execute any kind of motor plans. Because the idea with the motor system is that we are actually perturbing the system. We are causing the system to go from rest, which is very stable, to movement, which is highly unbalanced. And so when we move, we need to be very careful about our plan, our execution, and our correction of movements. And that's often based on the sensory systems. So obviously we can't really understand the motor system until we understand the sensory system first. So that's why section 2 covers the motor systems. And then the last system are these association systems. And these association systems are neither sensory or motor in structure. They're actually combinatory. Their goal is to integrate or connect the sensory information with the motor system to perform what we call higher order functions. So some of these higher order functions we've talked about in other classes: executive function, things like academic skills, performing increasingly complex abilities. And one other thing that's really important in the association system is just paying attention. So your attentional resources, allocating those attentional resources to what's easy to do and what's harder to do, so that you can perform each of these different actions seamlessly and efficiently. Okay, so we're going to go through a quick and dirty example or a run through of examples in which the different levels that we've talked about so far can be used, how they can be helpful for our understanding. Again, the subsequent chapters will go into a much deeper explanation. Not going to go into that. Okay, so I'm really not worried about those details at this moment. Okay, so the genetic level of explanation really focuses on the product of gene expression. That is the most important thing that we need to consider when thinking about genetics at all. I mean, for some of you guys that have not really had a lot of anatomy and physiology or have not had a lot of genetics, there's like in the popular or lay understanding of genetics, you have these genes and they don't really change. And if you have these genes, you're at risk or not at risk for some type of disease. That's really not how genetics really functions or works. Because what we know now is that genes are expressed. And not only do the coding regions of the DNA sequences matter for that gene expression, but we also need to understand how regulatory components of the DNA affect whether or not something will or will not be coded, and the extent to which it's coded correctly or not. So there are two regions, right? The coding DNA sequences that actually have, that are comprised of exons that serve as a template for mRNA, which eventually turn into or translated into proteins that create different things. They create microtubules that are important for axons. They create neurotransmitters. They create different organelles within the system to help make sure that the cells actually work. And again, the regulatory DNA sequences help us to understand how much, when, and how these coding regions actually do get expressed. And again, this idea of regulatory DNA as influencing the system is what we term as epigenetics. So the term genomics and neurogenomics is really the study of the entire DNA sequence, both the coding and the regulatory components, and how they affect the ultimate expression in terms of structure and in terms of function. Okay. One thing that I think is really cool in terms of genetics is that there are a lot of genes that are expressed ubiquitously, like across all different types of cells and tissue types. So we do see cells that are genes that are important for growth, for hypertrophy, hyperplasia, cell maturation, those types of things that are expressed across all tissue. And then we see a bunch of genes that are actually really unique to the brain. And unique to the way in which the brain cells function and the way in which the proteins are expressed, the way that they're important for synaptic transmission, which is really unique to the brain. So this Venn diagram kind of shows the overlap in terms of different types of genes, how they're expressed and where they're expressed. And then on the left-hand side, these are dots that represent expression levels of particular types of genes. And we just know so much more now based on postmortem samples in the developing the adult brain that different kinds of genes get expressed in different areas at different levels and at different times. And so it's kind of nice to see what that looks like overlaid onto the brain. And in this case, the color coding scheme is blue is like low levels expression, and then orange and red are high levels expression. And yeah, you can see that there are different samples that are taken across the different brain regions. And again, different genes are expressed differently. If you're interested in the genetic level and specific genes in particular, you can go to the Allen Brain Atlas. And there are examples from mice, and there are examples from developing fetuses, and samples from across the lifespan. And then there's also, you know, typical adult samples as well. But the Allen Brain Atlas is really awesome, and it shows you not just the structure and the function, but also provides the mRNA expression levels for different types of genes. Okay, so we will talk a little bit about different types of genes and how they often work together to help develop the structure and function of the brain. Oftentimes, we're going to see examples of genes in which, disorders in which a lot of genes contribute to the outcome phenotype. But this is an example of a single gene mutation, and this is causing microcephaly. You can see that the brain of this individual is much smaller. You can also see that it has less gyrification. You see it's less packed together. And again, this is the result of a single gene mutation that is associated with the development of the brain in this particular individual. So as I said, there are examples in which a single gene or a small number of genes are affected. The example that we'll talk a lot about is Huntington's disease. So that's an example in which a specific region with, or sets of regions within the brain are show degeneration. And actually it's a very, very fast degeneration. And it's highly heritable, and it's due to a very small number of genes. Most of the genes that most of the disorders we talk about are polygenic, right? A lot of different genes contribute. The expression of those genes or the disorders and regulation of those genes can contribute to things like Alzheimer's disease, Parkinson's disease, autism. And because most of the disorders that we encounter involve a whole bunch of different genes, the relationship between genotype and phenotype, right? Specific genes and specific behaviors is incredibly complicated, and it requires a lot more, not only a lot of participants, but a lot more information is needed to really understand these relationships, especially across development. And so we're still, I honestly, especially as a developmental neuroscientist, there's still a lot that we need to understand about how genes function and how they're expressed across the lifespan, especially in these different polygenic disorders. Okay. Most of what we know about neuroscience, and many of the Nobel Prizes that have gone in physiology have been due to our understanding of the nervous system through what are called model organisms. And model organisms are the comparative use of other organisms to understand basic neural functions, and then relating those functions and those structures to other species. And so we won't talk a lot about this in the class, but it's good to, as like a background, to understand why these things have been studied and how they've been studied. So in chapters 2 and 3, they talk a lot about axonal transmission and how synaptic potentials get transmitted across the axons from one cell to another. And a lot of that work was done by Hodgkin and Huxley in 1939 and 1952. They were awarded the Nobel Prize in 1963 for their work on the giant squid axons. One of the reasons why they were able to do this is the giant squid axon is visible by eye. So you don't actually need these high density microscopes to be able to see how those cells are actually working in real time. So that was a major thing, talk about Hodgkin and Huxley models throughout the class in terms of synaptic transmission. Another set of model organisms that have been used are cats. And cats have been used extensively to try to understand visual dominance columns and how the two eyes contribute to a sense of depth as well as space. And Hubel and Wiesel did a lot of this work in 1962 and 1968. And then they received the Nobel Prize in 1981 for their work in this area. And that actually gives us a lot of information about degeneration as well as reorganization, because you can actually patch the cat's eye or close one of their eyes, see how the visual dominance columns develop, and then lesion their eye or whatever, like release the eye, and then you can see how they reorganize. And it's really eloquent work. And then some of the stuff that's a little bit more recent is understanding basic sensory motor reflexes and learning by Eric Kandel. And this was really in the 1970s, 1980s, and then they received the Nobel Prize in 2000 for their work with aplysia, which is a sea slug that has a little tongue. It's a little siphon at the end. And what they did was they would stimulate the tongue, which would cause a sensory motor retraction or propulsion in response. And then over time, they would show that even this simple organism, this sea slug, was able to learn or pair a particular kind of stimulus to a specific kind of response. Okay, so that's some examples of model organisms for comparative neuroanatomy. The goal of comparative neuroanatomy is to compare one organism with another organism. So giant squid axons to humans. There are other examples in which we use model organisms that we really can't make comparisons with. So an example of that is bat or dolphin echolocation. We just simply don't have the sensory and cortical structures that are underlying those types of functions, but they're still important for us to understand in terms of understanding spatial awareness or understanding auditory processing. So these special types of species that have these unique sets of sensory and motor apparatus can be used under the umbrella of neuroethology. Okay. And then of course, the last set of organisms that are really helpful relate to genetic understanding of different types of expression. So we can use things like model organisms that have very simple genomes, so they can be sequenced really easily. So we see things like, this is an example of a C. elegans. It's a nematode, Drosophila, fruit flies, zebrafish, mice, rats. There's tons of examples in which we're able to look at those animals compared to what our system is like. And then we can also have transgenic animals in which we knock out or add in or knock in a particular sets of genes and see how they respond in terms of the outcome behaviors and the outcome anatomy and physiology. Okay. Let me take a break real quick. I'm gonna stop my share. Okay. See a show of hands. Okay. How are you guys feeling? I know you guys haven't seen the material yet. I'm not asking for a lot of depth. Okay. So let's just cover a couple things. So just to recap. Oh, what am I doing? Hold on. I'm losing you guys. Okay. So to recap, we covered some aspects of genetics. Key take-home messages in terms of genetics. 1, we don't just care about the individual genes, We care about the regulation of those genes, which result in the actual expression and development of different proteins that are going to be useful in terms of brain structure and function. We care about genes because there's a whole bunch of genes that are really specific to the nervous system and provide a lot of insights about the function of the nervous system. So things like genes that are important for synaptic transmission, genes that are important for developing neural connections between different brain regions, genes that are important for developing cortical structures, like the convoluted or cortical structure that we see. We care about genetics because there are lots of different diseases that can be caused by a single gene or a bunch of genes, and understanding those diseases helps us to really determine the extent to which those specific genes relate to the phenotypes that we see in terms of behavior and also in terms of anatomy and physiology. And the last bit that we care about in terms of genes is that we can use these different model organisms to understand, again, the effect of particular genes and expression of different genes with respect to the function and structure of the nervous system. Okay, that's the level of understanding I hope you guys get. Okay, okay, so we have 15 more minutes, and we're going to spend most of that time talking about the cellular level. Okay, so we're going to talk about the different kinds of cells. We're going to talk about how they look, what they do, and why the actual physical structure of those cells gives us insights about what their functions are. Okay, we're probably not going to get through the whole material, but that's totally fine. Okay, so wait, before I do anything else, questions about what I've covered so far? What I'm going to try to do for Wednesday is have a quick quiz, like one or two question quiz for you guys to take. It's not graded, on the genetic level and the cellular level before we transition to the structural level. Okay, just to make sure we're all on the same page. Okay. Okie doke. Let's transition. Okay, so the cellular level. What we care about here is the cells, right? We care about what cells are present within the nervous system and what are their organelles or components that are important and maybe somewhat specific to these types of cells. So there are two kind of categories, main categories of cells in the central nervous system: neurons and glia. Really up until the 2000s, everyone focused on neurons. They thought neurons were the most important thing and that glia were only support structures. They're not that important. What we know currently is that glia are fundamentally important to the way that the nervous system functions. And they're fundamentally important in terms of promoting repair and neural regeneration. Okay, glia are super, super important. So glia provide signaling for when there's need for repair given damage. They provide neurotrophic factors that promote growth and sustain cells. They also help to signal when there's been uncontrolled growth, like in cancers. And when glia don't function well, we end up getting these malignancies that cause tumors to grow within the brain. So again, neurons and glia, they act in concert together. Although neurons are important for actually creating the electrochemical transmission and electrical potentials, the glia are really important for their overall function and whether or not they're going to survive and be successful. We tend to see some of the basic organelles that we see in other cells, so mitochondria, endoplasmic reticula, and different types of proteins that are important for developing structure. But what's interesting is that because the cellular structure of neurons in particular is kind of weird, they're not like, spherical. They're not these small structures. They're super elongated and they have a very weird morphology. We find that the organelles are distributed differently and where they're most needed. right? So mitochondria are really important for ATP and for having energy. It's really necessary near the synapses, where you end up having transmission along those synapses, reuptake of different neurotransmitters, and packaging of different organelles. And so the mitochondria we tend to find not only in the cell body near the nucleus, but also in higher density in the synapses or near the synapses. The endoplasmic reticulum, which is going to be really important, right, for creating proteins, are going to be found near the nucleus, because obviously we have translation of mRNA into proteins. And so the endoplasmic reticulum is necessary and to be near the nucleus. And then the fibrillar and tubular proteins are going to be found within the tubes and fibers within the axons and dendrites. And what you'll find is that the cell itself, the neurons themselves, are really dynamic. There are some really interesting YouTube videos of dendrites actually extending out. And you see these little buttons are called boutons that extend out from the dendrites that actually literally reach towards some type of neurotransmitter or some other type of chemical that's in the cytoplasm, and kind of reach out towards them. And that's really important for helping to build those connections between dendrites and axons. So it's really important for those building block proteins to be near those areas that might require a little bit more scaffolding. Okay, so here's an example of our like basic schematic. This is going to be a cortical pyramidal cell. You can see that it kind of looks like a pyramid. The center of the cell is going to be the cell body, or the soma. And you'll see 3 different processes that extend out from the soma. Those are all dendrites. There's one that's on the top. It's called the apical dendrite. And then you'll have 2 on the outsides. Those are lateral dendrites. That creates opportunities for cells to interact, not just from an axon to interact with the apical dendrite, but an axon can interact with any part of those dendrites, and including the cell body. You'll see that the nucleus of the cell is surrounded by ribosomes and endoplasmic reticula. And then at the base of the soma, you'll see the axon hillock and the descending axon. So these are some of the major components. But what's important is that these cells are organized in columns, and you tend to find different parts of the cell in different layers or levels within the cerebral cortex and within other areas of the brain. And we'll see what that general organization is later. But for the time being, we just need to know that there are certain, there's a certain structure of this particular type of cell. These pyramidal cells are found mostly within the cerebral cortex and what we call the forebrain. And then there are different types of cells that are found in other parts of the central nervous system. And again, the structure or the morphology of the cell gives a lot of insight as to how that cell might operate. So for example, with the pyramidal cells, if you have multiple pyramidal cells organized in like kind of in tandem, you can have this cell or, communicating with this cell. You can have this cell communicating with this cell. So it enables lots of different networks that can be created kind of horizontally, vertically, and in depth. So it's kind of nice to see how it's orchestrated. Okay, so let's look at other cells. These are other examples. These are stained micrographs. Here we see the axon of the cell, and then surrounding that in that kind of yellowish is the myelin structure. Myelin is basically oligodendrocytes. So they are fatty tissues that surround the axon. You'll see that they don't surround the entire axon. There are breaks. We'll talk about what those are called in a moment. So this one is the axon and the myelin. These are cross-sectionals. So imagine just cutting here, right, where the arrow is. We'll see that the cells are kind of bundled in tandem. Again, you can think about it as a series of pipes. And then again, surrounded by myelin And the myelin actually coils around. And so when you have the development of a myelin structure, you'll have multiple layers developing around, and that increases the axon diameter overall in terms of this cross-sectional area. Okay. Okay, so dendrites. Dendrites can interact with each other. They, again, can take the form of an apical dendrite. We'll see a couple other different types of arborizations. Again, this is all taken from pyramidal cells in the cerebral cortex. Here is an example of a cell body. Again, there's an apical dendrite. There's one of the lateral dendrites. Hold on a second. And then here's an example again of our myelinated axon with, so axon with myelin. And you can see that there's a break in that axonal sheath, and that's called the node of Ranvier. And we'll talk about why that's important for what's called saltatory conduction along the synapse. Okay. Let me just see where we are. Okay, so these are a bunch of different examples of different types of morphology of all of these are neurons, but they all look super weird, okay? So the example that I showed you was a very simplified schematic of a cortical pyramidal cell. This is a much better example of what those pyramidal cells truly look like. Again, we have a cell body in the middle. We have an apical dendrite that extends up through different layers within the cerebral cortex. We have lateral dendrites that extend out laterally from the cell body. And then we have a descending axon, but we have what are called axonal collaterals, so other branches that can create these recurrent networks, both with itself and with neighboring cells. So that's an example in the cerebral cortex. In the retina, we have cells that are called bipolar cells. So they have dendrites on one end. The arborization of the dendrites is relatively limited, meaning that it doesn't make a lot of connections with its neighbors. It has very specific cells that it connects with. And then it has a single unbranched axon and has a very small axonal arbor at the end in terms of the number of terminals. So with this retinal bipolar cell, it receives only a few inputs and it only sends a few inputs or outputs. And so, again, that looks very different than what we see with the cortical pyramidal cell. Cortical pyramidal cells get a whole bunch of inputs from neighboring cells and can send a lot of outputs. The retinal ganglion cell, which is another cell that's within the visual system, takes in a lot of inputs. You can see that the dendritic branching is quite extensive. It will connect with lots of cells that are organized to synapse directly on top of that, that dendritic branching, but it only sends a single axon. So it receives a lot of information, collects that information and sends a single axon and does not branch much. And a weird cell, again, within the retina is what are called retinal amacrine cells. These are supporting cells. It doesn't have an axon. Okay, so there are cells that don't actually have all three components of our traditional neuron. So it has a pretty elaborated dendritic branching and it only has a cell body. There is no axon. And then we can see, well, this is my favorite cell, the Purkinje cell. This is within the cerebellum. So let's get an idea of what this cell does. So all the green here is a dendrite. And there is a single axon, but it has one single collateral that reaches back through its own dendritic branches. So it receives a ton of input from neighboring cells organized in parallel, as well as other cells that are called, it's called interdigitate. It goes in between, like it will have its branches like this, like a tree, and then there's cells that interdigitate and are organized perpendicular to those cells. So it can create these really small networks just within the dendritic branching. It'll collect all that information within the cell body and send as a single output. So this is really interesting in terms of, again, the input-output structure based on the morphology or the structure of those cells. Okay. I'm going to go ahead and end here. Take a deep breath. This is good. We will continue talking about the cellular level of description. We have a few more slides to get through, and we'll talk about take-home messages. And then we'll do a short quiz just to make sure we're all on the same page. Let me stop sharing my screen. Okay, cool. So what I would like you guys to do is, again, go ahead and download the textbook from the Canvas site. Start watching the material that's posted for that first section from the University of British Columbia's neuroanatomy site, and continue reading chapter 1. We will get through chapter 1 next time. I'll update the syllabus so you have it there. And let me know if you have questions as you're reviewing the material. Okay, I'm going to stick around for just a little bit longer. I will post this recording onto Canvas as soon as we're finished with class. Okay, thanks guys.